CRISPR, clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing technology that allows scientists to precisely modify DNA sequences. This innovation has transformed genetic engineering, with profound implications for disease treatment, agriculture, and fundamental biological research. 1. Natural defense mechanism. CRISPR was first identified in bacteria, where it serves as an adaptive immune system. Bacteria use CRISPR sequences to store snippets of DNA from viruses that have previously attacked them. If the same virus attacks again, the bacteria produce RNA segments from the CRISPR sequences to guide the Cas enzymes to the virus's DNA, cutting it and neutralizing the threat. This natural defense mechanism was discovered in the late 1980s and early 1990s, but its potential for gene editing wasn't realized until the 2010s. 2. Cas9 enzyme. CRISPR technology relies on the Cas9 enzyme to cut DNA at specific locations. The Cas9 enzyme is guided to its target by a guide RNA, gRNA, sequence that matches the target DNA sequence. When the Cas9 gRNA complex binds to the target DNA, Cas9 makes a double-strand break. The cell's natural repair mechanisms then kick in, which can be harnessed to introduce specific genetic changes. 3. Correcting genetic defects. The CRISPR-Cas9 system has successfully corrected genetic defects in embryos, plants, and animals. In human embryos, CRISPR has been used experimentally to correct mutations responsible for genetic disorders such as beta thalassemia and hypertrophic cardiomyopathy. In plants, CRISPR has been utilized to develop crops with desirable traits such as disease resistance and enhanced nutritional profiles. In animals, gene editing has been used to create models for human diseases, aiding research and potential therapies. 4. Gene drives. CRISPR could be used to create gene drives, spreading genetic modifications through populations. A gene drive increases the likelihood that a specific gene will be inherited, allowing it to spread rapidly through a population. This has potential applications in controlling disease vectors like mosquitoes, which transmit malaria and other diseases, or in eliminating invasive species that threaten ecosystems. However, the ecological impact and ethical considerations of releasing gene drives into the wild are significant concerns. 5. Ethical concerns. CRISPR raises ethical issues such as off-target mutations and the prospect of designer babies. Off-target effects occur when CRISPR edits unintended parts of the genome, potentially causing harmful mutations. The idea of designer babies, where embryos are edited for non-medical traits like intelligence or appearance, raises profound ethical questions about human enhancement, equity, and the societal implications of such technologies. Regulatory frameworks and public discourse are essential to navigate these ethical landscapes. 6. Cancer treatments. CRISPR is being explored for developing novel cancer treatments. One promising application is in immunotherapy, where CRISPR is used to engineer T cells to enhance their ability to target and kill cancer cells. For instance, CRISPR can be used to create CAR T cells with improved precision and effectiveness against specific types of cancer. Researchers are also using CRISPR to identify and validate new drug targets and to understand the genetic mutations driving different cancers. 7. Agricultural applications. CRISPR can enhance crop yield, pest resistance, and nutritional content. By editing plant genomes, CRISPR can create crops that are more resistant to diseases and pests, reducing the need for chemical pesticides. It can also improve crop resilience to environmental stresses, such as drought or poor soil conditions. Enhancements in nutritional content, such as increasing the levels of essential vitamins and minerals, can address malnutrition and food security issues globally. 8. Malaria control. CRISPR has been used to modify mosquitoes to resist malaria parasites. By altering genes in mosquitoes that are essential for the malaria parasite's life cycle, scientists aim to reduce the ability of mosquitoes to transmit the parasite. For example, the gene modification can make mosquitoes less hospitable to the parasite or sterile, reducing their population over time. Field trials and ecological studies are ongoing to evaluate the effectiveness and safety of this approach. 9. Viral infections and genetic disorders. CRISPR-Cas9 is being researched for treatments of viral infections and genetic disorders. In the case of viral infections like HIV, CRISPR can potentially excise the viral DNA from infected cells, offering a potential cure rather than just suppression. For genetic disorders, CRISPR offers a way to correct mutations at the DNA level. Research is particularly advanced for diseases like Huntington's, where a single genetic mutation causes severe neurological damage, and Duchenne muscular dystrophy, which leads to muscle degeneration. 10. Patent battles. CRISPR's discovery has led to significant patent disputes. The primary dispute has been between the University of California, Berkeley, and the Broad Institute of MIT and Harvard over who first invented the CRISPR-Cas9 technology for gene editing. These legal battles highlight the immense commercial potential of CRISPR-based technologies, which could lead to significant economic benefits for the holders of foundational patents. The outcomes of these disputes will influence the direction of research funding and the accessibility of CRISPR technologies. Conclusion: CRISPR and gene editing are at the forefront of biotechnology, promising revolutionary advances across multiple fields. As research continues to progress, it is crucial to address ethical concerns and ensure responsible application to harness CRISPR's full potential for the betterment of society and the environment.